Now in today's video we are going to discuss about retina, and retina is this layer of the eyeball. It is the interior most layer of the eyeball, and it is the layer or the neuronal layer which converts the light that is coming out from the outside into the neuronal signals. Now we have certain things. First of all, when forebrain develops, it has an optic vesicle formed, and that optic vesicle is connected to the forebrain. If this is the forebrain, there is an optic stalk, and then we have the optic vesicle. Now this optic stalk, it changes into the optic nerve, and this double walled membrane or something like that, this double walled cup shaped cavity, it develops into the retina. And retina has two layers. The layer that is toward the choroid, it is known as the so now this layer of the retina which is towards the choroid, it is forming the pigmented layer, and the one that is towards the inside, it is forming one layer which is known as the neural layer. So towards the choroid we have the pigmented layer, and towards the vitreous body we have the neural layer of the retina. Now they are saying that this pigmented epithelium or pigmented layer of the retina, it is very much strongly adherent to this Bruch's membrane of the choroid, but it is not well adherent to this neural layer which is towards the vitreous body. And that is why when we do the histological sections, then the pigment layer which is towards the exterior and this interior layer, that is the neural layer, they are split, and we can easily have a distance between them. So this was the general overview about the retina. Let's move to its histology. So now these are the layers of retina that we need to know in this diagram, and we will be done with everything. Now I told you that on the exterior side towards the choroid, we had this retinal pigment epithelium, and this pigment epithelium has a strong connection with the Bruch's membrane, but it is not anatomically related to the neural layer of the retina. So we have the outer pigment epithelium and the neural layer of the retina, and both of them, they are not anatomically attached to each other, as a result of which the retinal detachment is a very common issue. Second thing is that this retinal pigment epithelium is very much strongly adherent to the underlying choroidal layer, which is the Bruch's membrane. Now this retinal pigment epithelial cells, let's discuss which type of cells they are. They are actually low columnar cells, as we can see, and they have a basal nuclei. Also they have apical portions, and these apical portions are towards these neuronal layer of the retina, and these apical portions are strongly laddered with large amount of these melanin granules, whereas the nuclei is on the basal side. Also it has a cytoplasm which is made up of the Golgi apparatus, the mitochondria, the smooth endoplasmic reticulum, lysosomes and phagosomes. And these retinal pigment epithelium cells, they are very much adherent to each other through tight junctions which are zonula occludens type. With that, we come to an end of the retinal pigment epithelium. From here, we are going to begin with these type of cells, which are the neuronal layer cells. Firstly, you need to know that how many cells are present in this layer. The number of cells, first of all, is these cells, which are the rods and cones cells. Then we have the second cells, which are coming as the bipolar cells, and third cells, which are the ganglion cells. And how they are arranged is the whole apparatus of this 10 layers. So first of all, we have the layer of rods and cones. First of all, we have the retinal pigment epithelium. After that, we have the layer of rods and cones. Then we have external limiting membrane. Then we have an outer nuclear layer, the outer plexiform layer, an inner nuclear layer, and inner plexiform layer, the ganglionic cell layer, and the nerve fiber layer. Then we have an inner limiting membrane. Let's discuss all of that. We are done with the retinal pigment epithelium. Now we will go to the layer of rods and cones. So first of all, we need to know that these rods and cone cells are giving their photosensitive processes, and these photosensitive processes interdigitate with the apical processes of the pigment epithelium, but they do not have a physical connection with them. That is what we can see in the layer of rods and cones. Then we have the external limiting membrane. Then we come to this outer nuclear layer. And this outer nuclear layer is further divided into an external and an internal portion. The external portion has only the cell bodies and the nuclei of the cone cells, whereas the internal layer, it has the cell bodies of the rod cells. So if we place a pencil or pen like this and we divide this outer nuclear layer into two portions, so the outer portion will have all the nuclei of the cone cells and this inner portion will have all the nuclei of the rod cells. Then we have the outer plexiform layer. So now these rod and cone cells, they're going to give their axons, and their axons are going to synapse with the axons of the second cells, which are the bipolar cells. Now this synaptic layer, it is known as the plexiform layer. And since we have two synapses, so the outer synaptic layer is known as the outer plexiform layer. Similarly, as you know, these bipolar cells will also have their neurons. So we have another neuronal layer which is known as now inner nuclear layer, and this nuclear layer has the nuclei of the bipolar cells. There is no division there. All the nuclei that we can find in this layer, they are of the bipolar cells. But now again, these bipolar cells are going to give their axons, and they provide an inner plexiform layer. And this inner plexiform layer has now the synapses of the bipolar cells with the ganglion cells. So with that, we come to an inner layer of the nuclei, and now we call this the ganglion layer. So we had three nuclear layers. One was the external nuclear layer or outer nuclear layer that had the rod and cone cells. The most exterior portion had the cone cells, and a little bit towards the interior we had the nuclei of the rod cells. This layer, the inner nuclear layer, had the nuclei of bipolar cells, and this layer, it is the ganglion layer. Now these ganglion layers or the ganglion cells, they are going to give a large number of the axons, and their axons are going to form the outer or optic nerve fibers. And then we have the inner limiting membrane. With that, we have the all 10 layers of retina covered. This was the easiest thing to be studied. We just need to know that the cells, how many cells are there? There are three types of cells. There are the first cells, rod and cone cells, the bipolar cells, and the retinal cells. Now, we also have first, second, and third order neurons in the retina. So these are rod and cone cells. They are actually forming the first order neurons. They synapse with the ganglion cells, and they synapse with the bipolar cells, and then we come to the second order neuronal layer, and then they synapse with the ganglion cells. The bipolar cells synapse with the ganglion cells, and they form this layer which is known as the third order neurons. And from the third order neurons, their optic nerve fibers are produced. With that, we come to an end of the retinal histology. Now we are going to consider the functions of retinal pigment epithelium. This retinal pigment epithelium. First of all, we have the first function of the retinal pigment epithelium as the absorption of light. As I told you that these retinal pigment epithelium, they had like certain melanin granules. So what is going to happen? A large amount of light is going to come, and only bits of light will be absorbed by these rods and cone cells. The remaining light will be absorbed by these melanocytes, and when the melanocytes, they absorb the light, they produce darkness in the eye. That is why dark chamber effect is produced in the eye. Now second thing is the storage of the vitamin A, and it's applied to the photoreceptors. Now what happens is that, as I told you, let's come to this layer. This plasma membrane of the apical surface of the retinal pigment epithelial cells, this plasma membrane is richly supplied with a large number of vitamin A. And the interchange of the vitamin A between these photo receptor processes of the neuronal cells or the rod and cone cells and the plasma membrane, this interchange allows us to have the walled visual cycle of the vitamin A. Now, when we were discussing the retinal pigment epithelial cells, I told you that they have a large number of phagocytes. Why are there a large number of phagocytes present over there? Because these phagocytes are going to help to absorb the lamellar layer of the photoreceptor cells. Whenever the lamellar layer is shed off, that lamellar layer, it is eaten or it is scavenged by the phagosomes of the retinal pigment epithelium. Now, I told you that there were between these low columnar and retinal pigment epithelial cell, there were a lot number of the tight junctions, and these tight junctions were of the zonula occludens type. And what they did, that they blocked or sealed all the intercellular space. So the only method for the movement of the gases, water, nutrients, metabolites from the external capillaries to the interior layer of the retina is only through these retinal pigment epithelial cells. So they make the blood retina barrier. Also, they have a supportive or nutritive role, is that they're going to themselves form the ATP because they have large number of mitochondria, and also provide with growth factors because they have a large number of Golgi apparatus as well as the smooth endoplasmic reticulum. With that, we come to an end of the retina histology. In the next video, we are going to discuss about the elements of neural retina.